So hi there, my name's Cameron Brown, and today I'm going to talk to you about the development of microfactories for pharmaceutical manufacturing, CMAC, as well as some thoughts on the outlook of this area of research. So to begin with, it's worth recapping what we mean by a microfactory. In our case, we've defined them as lab-scale platforms that integrate multiple unit operations to deliver the ability to manufacture a drug substance or drug product. And critically, these platforms can control, measure, and optimize the transformations across multiple length scales from molecule, crystal, and particle. So to develop a microfactory, we've envisioned a workflow where, given a molecule, we would identify the transformations required and associated kinetics. Based on this, we would then select from a library of unit operations and equipment that would be optimal for these transformations. And each entry in the unit operation library would be pre-characterized to identify its operating window and would be accompanied by a simulation model. And then finally, these unit operations would be integrated into a single platform with an overarching control system. So with that in mind, what are the developments in microfactories that we've achieved to date? So back in 2014, before the actual CMAC hub, through RPF funding, we initiated the process for the design and construction of skid-mounted modules for continuous crystallization. And these were built and designed in conjunction with Micronova, NETEC Solutions, AWL, Perceptive Engineering. And these gave us 5 unique modules and associated ancillaries to be able to deliver continuous crystallizations robustly and routinely. Later in the same year and into 2015, in partnership with AstraZeneca and Perceptive Engineering, the MOPS project came to conclusion, which demonstrated a flexible control system on 2 different crystallization platforms, and crucially with the ability to dial a particle size using feedback from PAT. And into 2015 itself, almost a year after the first design, we had the commissioning of our skid modules, and they were demonstrated at the Open Day that year. And these skid modules were then used again in 2016 amongst other equipment as part of the final proof of concept stage for our workflow for seeded cooling crystallizations, where the skids were really critical in delivering 3 different sizes of API by first intent. Also that year, our process control infrastructure expanded with help from Siemens and Perceptive Engineering with the deployment of the Siemens PCS7 and SciPac management systems. And this was demonstrated on our in-house built COBC, and this gave us an industry standard control system with flexibility and room for expansion. So up until this point, all the unit operations have been operated independently. However, in 2018, we had our first demonstration of taking material from a continuous crystallization platform and transferring it into a continuous filtration platform. The theme of unit operation coupling continued this year with partnership with Perceptive Engineering and Process Systems Enterprise as part of the DOT project. We demonstrated the ability to use a twin screw extruder as a feeding device to dissolve slurry before feeding into a single stage MSMPR for crystallization. This demonstration also brought about the ability to control over multiple unit operations, as well as our expansion of linking control and process modeling. And also that year, we set about the design and construction of a custom continuous stirred tank platform that would be flexible in terms of the number of stages and volume of each stage, as well as the ability to transfer material between different stages through different methods. And staying in 2018 and in time for the Open Day that year, we demonstrated perhaps our most integrated and intensified process to date, which included a continuous crystallization, a spherical agglomeration, and filtration within a single fume hood module. And early in 2019, we demonstrated perhaps a unique capacity of the microfactory modules. As they're inherently small and compact, they can be essentially put in places that traditional manufacturing equipment can't. And so in this case, a hot melt extruder was modified and taken to the diamond light source synchrotron. And so for the first time, we could get advanced in situ measurements as extruded left the extruder nozzle. We also built upon some of our previous developments with our custom container stir tank platform being upgraded to work with the Siemens PCS7 and SciPac management systems. And through PharmaNV and GPROMs from Prescriptive Engineering and Process Systems Enterprise respectively, we've established a three-way link between the hardware control system and the process model. And this allows the model to aid the controller, allows data to be sent from the hardware to parameterize the model, and for the controller to be used virtually for training purposes. And on to 2020, and certainly for a majority of research this year, it's been obviously upset by COVID and the pandemic. However, very early this year, we did demonstrate another case of process coupling with our modules. And this included the synthesis and crystallization within the single continuous stir tank platform, as well as isolation into the filtration platform. And that brings us up to where we stand today. So in summary, we now have a unit operation library that covers synthesis steps and workup through liquid extraction, versatile crystallization platforms that operate in a range of modes and deliver different particle engineering techniques, wet milling, spherical agglomeration. We have a filtration platform for our drug substance isolation. And into the drug product space, we have feeder and blender platforms that could be linked to extruders or tablet press, but also platforms to deliver complex structured drug products through 3D printing. And to facilitate the coupling of these unit operations, we have a process control infrastructure that allows for the management, monitoring, recording of control hardware and PAT, but also allows for the two-way communication between our process models and our control systems. And then finally, to bring all this together, we have workflows which allow us to develop and characterize our processes in a systematic manner. And as with our unit operation library, these span everything from crystallization through to drug products. And as I touched on earlier, these workflows help us understand a molecule and particles transformation and the equipment or processes necessary to achieve that. So one area that's still open for discussion is how microfactories could be deployed, and this is both technology and business case dependent. And if we look at traditional development pathway, experiments would be performed at increasing scales from lab to technical and pilot and production, with the process undergoing validation each stage. Perhaps what we could do is that we could replace the technical stage with a microfactory, where we have a lab development and then we develop a microfactory. And then that microfactory is either scaled up straight to production, or it can be numbered up, avoiding increasing vessel sizes, equipment sizes. Or the final approach is that we could develop straight into the microfactory. And so actually here we have very little scale up or tech transfer routes, but perhaps do use more material during lab scale development than we do traditionally. So very quickly, just to sort of wrap up and give some views of what's happening in future, what some of the outlook is. If we compare the drug product processing space to drug substance, we see a fewer number of different types of unit operations. And certainly there's a desire to move towards simplified unit operation chains, such as direct compression. Could we do the same thing in drug substance space? So for less variety in unit operations, or even a fixed unit operation chain, how many molecules could we develop a process for, and even if it wasn't the optimal configuration? I touched on lab scale development and the notion that microfactories were like a scale above this, but what if the microfactory concept extended back to lab scale, and we had automated and highly instrumented what we could call data factories, which would give us all the required kinetic information and model information that we need for a given transformation. There's some practical challenges still exist. For example, ancillaries for microfactories are often overlooked, and these can take up as much room and require as much consideration as the microfactory themselves need. And how we deal with ancillaries will depend on how we look to deploy it as well, if it's numbered up or scaled up, for example. And then lastly, we can look at equipment designs itself. So there are still room for optimization. Pharmaceutical equipment hasn't changed since its first inception. However, with advanced manufacturing and design techniques, we can potentially build equipment that offers measurable advantages over current designs, and start to look to things like inherently incorporate PAT and sensors, so our monitoring is much closer tied to our process. So hopefully that's given you a highlight of the microfactory work at CMAC, from the unit operation library that we've built to its underpinning control system and the workflows we've developed to aid in their development, and the capability this all brings, to the future of pharmaceutical manufacturing. And hopefully you've seen as well some of the challenges that we're looking to address in the direction of the future research. So thank you to everyone that helped contribute information to this talk today, and thank you for your time to listening today as well.